All right, class, we have focused on T cell activation. Now let's focus on the activation of our B lymphocytes, our B cells. Our B cells, generally speaking, are going to be activated by a T helper cell. So we need to have a T helper cell go to the B cell and activate it. That T helper cell is going to have its CD3, CD4 molecules helping to bind to the antigen, and it will present the antigen to the MHC receptor on the B cell. So the T helper cell will expose the B cell's MHC class II receptor to the antigen, and once that B cell's MHC class II receptor is exposed to the antigen, then our B cell becomes activated and begins to differentiate. And our B cells differentiate into three primary types. There'll be B regulatory, memory B, and plasma cells. B regulatory cells are going to then travel to T cells and trigger replication among the T cells. So B regulatory causes there to be more T cells. Our B memory cells are long-term cells that are going to give us immune system gives us long-term immunity to a pathogen. And then finally, those plasma cells are going to go through many, many, many rounds of rapid reproduction and start to produce antibodies. The main focus that I want you to focus on for B cells is that B cells, when activated, turn into plasma cells, which then produce lots and lots of antibodies. We get our antibodies primarily from plasma cells. So if we look at our antibodies, they're going to have a Y-shaped arrangement. So a generic antibody is shaped like the letter Y. And it will have a couple different regions to it. There's going to be the antigen binding fragment, the FABS. This antigen binding fragment of the antibody is going to have a specific fold and groove that will accommodate the specific epitope of the antigen. Remember, the epitope is the part of the antigen that the antibody actually sticks to. We also have the FC, the crystallizable fragment, named because you can cause it to precipitate and form crystals. And then we're also going to have some heavy chains and some light chains. And within our antibodies, we have our FAB, FAC regions that are going to include some light chain, some heavy chains, and then some swiveling of the FAB fragments. This slide doesn't actually make a lot of sense. This slide makes a lot more sense. So let's focus on this slide. I'll repeat myself. We have our heavy chains. The heavy chains are long chains. Then we have the light chains. The light chains are the short chains. So they are the ones that don't have as much mass. They're the light chains. Now within our light chains and our heavy chains, we have variable regions. These variable regions change from antibody to antibody. And within the variable region, we're going to have antigen binding sites. So each antibody is capable of binding to two antigens. This variable region up here is bound on the FAB, and it's worth mentioning that the FAB can swivel around its axis. So if we were to draw a dotted line right down the middle, this antibody could spin on its axis, and the FABs can swivel around each other. We have disulfide bridges holding the fragments of our antibodies together, giving us some anchoring points between the light chains and the heavy chains of our antibodies. Oh, there goes my eraser. The FC, the crystallizable region of our antibody, is going to be constant across the antibodies. This FC region is going to be the same regardless of what antigen can stick to the variable region of the antibody. We also have a hypervariable region of our antibodies. This hypervariable region is the site on the antibody specifically where the epitope binds. So if we're to go back a slide here, our hypervariable region is this little chunk right there within the variable region, the specific spot that binds to the antigen. This amino acid content of our content of the hypervariable region, as we would assume, since it's hypervariable, is extremely varied. And that makes sense. The specificity of the antigen binding sites in our hypervariable region is very similar to the specificity for an enzyme and a substrate, kind of like that lock and key model we talked about earlier on in this course, where we talked about how a specific substrate fits in a specific active site of a specific enzyme. the specificity of the two Fab sites is identical. So if we back up here, the specificity right here for that binding site is exactly the same as the specificity of that binding site. So we can have the same antigen bind to either spot. So here we can see an excellent summary slide of antibody function. We can see the antibodies binding to the surface of bacterium. And if a bacterium is covered in antibodies, it's going to be opsonized. Opsonization is the process where we make a microbe ready to be phagocytized by a phagocytite. So if we take this object and cover it in antibodies, we make it more likely to be phagocytized. In other words, covering it in antibodies is opsonization. We can also have antibodies bind to viron particles, neutralizing the viron particles. We can have antibodies bind to separate bacterium, causing them to clump together. We can have antibodies bind to a bacteria and initiate the complement cascade. Recall, the complement cascade is part of our second line of defense. It's a generic mechanism that perforates the cell envelope. Or we can have antibodies bind to and neutralize proteins that are secreted. So let's talk about some healthcare applications from this slide. If you've been exposed to a virus and developed immunity for that strain of virus, you will now have antibodies present in your body that can bind to and neutralize additional viron particles. If you have received a toxic bite, let's say you're bitten by a poisonous spider or a poisonous snake, you've been bitten by some kind of animal or creature that has toxic venom, antivenom, generally speaking, is going to be a concentration that contains antibodies which bind to the specific toxic proteins that were injected into your body, neutralizing those toxic proteins. That FC region, that crystallizable region of the antibody, is going to have an effector portion that binds to the membranes of our macrophages. The effect of binding an FC region depends on our cell's role. So if we have opsonization, the attachment of antibodies to foreign cells is followed by our phagocytes. So opsonization is antibodies binding to a cell, causing the cell to be phagocytized. If we look at this FC region on immunoglobulin E, which is a specific kind of antibody, that FC region of immunoglobulin E is going to bind to basophils and mast cells and trigger an allergic reaction. There are multiple kinds of immunoglobulins. And just to make sure that I fully explain it, an immunoglobulin is also known as an antibody. Just like an immunogen is the same thing as an antigen. We have different kinds of antibodies. Here are the 5 different kinds of antibodies that have been discovered so far in human beings: IgG, IgA, IgM, IgD, and IgE. Let's talk about IgA. IgA, immunoglobulin A, is a monomer antibody that circulates in very small amounts in the blood. It's a dimer form of an antibody, so we'll have two monomers stuck together to form the dimer of IgA. And IgA is present in very high concentrations in the mucus secretions and in saliva and in breast milk. This dimer is 2 monomers that's held together by a J chain to facilitate the transport of IgA across membranes. This J chain that holds them together is hydrophobic in nature, which helps to aid in the transport of that antibody across a membrane. IgA is present in mucous membranes. It's in secretions of our body. We have lots of IgA in our breast milk. If we look at colostrum, the very first thick, fatty milk that's secreted immediately after a woman gives birth, colostrum has a very high concentration of IgA, and this coats the newborn infant's intestinal tract with antibodies. But there's more than just IgA. We spent some time talking about IgA, that dimer. IgA is not a very common antibody, though. It only makes up about 13% of the total antibody concentration of our body. The single most common antibody is IgG. IgG is a monomer antibody. It makes up 80% of the total concentration of antibodies in our bodies. IgG is very small. It is going to function primarily to opsonize bacterium in our bodies and to neutralize proteins or toxins that are in us, and to trigger the complement cascade. So because IgG is our classic antibody that triggers opsonization, the complement cascade, and will also neutralize toxins and virion particles, IgG is our most effective, most powerful antibody, and most common antibody. IgA is the one that's present in high concentrations in breast milk. IgM, immunoglobulin M, is the big fatty of the group. It's the antibody that's a pentamer. It has 1, 2, 3, 4, 5 subunits. This antibody makes up approximately 6% of the overall concentration of antibodies in our body. And this is going to be a B cell receptor antibody. IgM is on our B cells and serve as a B cell receptor to help mediate the immune response. IgM also functions particularly well when we want a bacterium to be clumped together. These 5 subunits are each capable of binding to 2 bacterium. So theoretically, an IgM antibody can cause 10 bacterium to get stuck together. IgD, immunoglobulin D, is exclusively a receptor on a B cell. One of the reasons why IgM, or one of the things that differentiates IgM from IgD, is IgM is produced first in response to an antigen. So the very first antibodies that are being secreted in response to that antigen are antibodies that can bind to incredibly high numbers of that antigen. And then at the end, we have IgE. IgE is present in very low concentrations. For a very long time, IgE, IgD, and IgG were all mixed up with each other because they're all monomer-based antibodies. IgE is from mast cells and basophils and is going to help mediate allergic reactions and remove parasites from our bodies as well. If we look at the concentrations of antibodies in our body with the primary versus secondary response, our primary exposure, our first exposure to the agglutinin or the antigen is going to initiate the primary response. This primary response has a latent period. It takes a while to activate our adaptive or specific immune system. After activating our specific immune system, we're going to have an initial rush of IgM. IgM is the first antibody on the scene that's being produced. After that IgM is produced, we're then going to have a rush of IgG that's produced. So during that primary response, we're going to have a high concentration of the antibodies. But it takes time. There's that latent period where we're priming the pump. But now, after that initial exposure for the primary response, we now have memory cells, B memory cells and T memory cells. And then the second time we're exposed, those memory cells are able to respond very quickly and almost instantly produce high concentrations of IgG and high concentrations of IgM. And this is going to be every subsequent exposure. The secondary response is harder, better, faster, stronger. In every fashion of the term, it's going to be more powerful than that primary response. And this is the goal of vaccination. We vaccinate somebody to initiate a primary response with a weakened or partial pathogen that doesn't have all the nasty symptoms, so that when they're exposed to the real deal, they'll have a secondary response with a much higher concentration of IgG and IgM, hopefully preventing them from getting sick. The concentration of antibodies in our blood serum is known as a titer. So a titer is the level of an antibody in our blood serum. Memory B and memory T cells are only created from clones activated by the specific antigen. So a memory B cell and a memory T cell can only respond to the specific antigen that activated their clones. New research right now in the field of immunology is showing that a particular antigen can result in memory cells that are chemically related to it, even if those antigens have never been exposed to the host, never been seen by the host. In other words, we're starting to find out that slightly different antigens, or one antigen can give us immunogenicity, or can give us immunity to a similar but slightly different antigen. The activation of our memory cells occurs when those memory cells activate, or recognize, I should say, the specific epitopes of the specific antigens that they are pre-programmed to recognize. This is going to potentially give us the use of non-pathogenic microbes in vaccines against the more dangerous microbes in the vaccine, if we could potentially find ones that are similar to, but not the same as, those pathogenic ones. So, concept check class. Which antibody type is matched with its description? You can go ahead and pause the video, check your textbook, check your notes, or you can rewind the video. Get me an answer. 1, 2, 3, 4, 5. IgG is the most prevalent antibody in our body. IgM is the first one that's produced. IgA is the one that's going to be secreted in our mucous membranes and breast milk. IgD is going to be in high concentrations as a receptor on our, let's double check right here, I want to make sure I get it right for us. IgD, boop, boop, boop. Yep, is our receptor. And then IgE is responsible for mediating allergic responses. If you have any questions, please post them to the discussion board, shoot me an email, or get back to me in my office. Happy studies.